animal cells can be cultured in laboratory on a medium containing appropriate nutrients for growth. Growth of cells is obtained by dispersing any animal tissue and inoculating it on an artificial medium. The animal cells are very delicate and can be grown on any laboratory medium with great difficulty. They require a substrate for attachment in order to grow. The cell growth obtained by disaggregating of the animal tissue on a culture medium is a primary culture, as already discussed in earlier module. Visible colonies from the cells placed in culture vessel develop within 10 to 14 days. On development of a primary culture, some cells may not survive on the artificial medium. Some of them may survive for a short period of time and eventually die, and some other may still survive in the culture for long period. The cells exhibiting prolonged survival may also show slowing down of growth due to reduced availability of space and nutrients. These cells are transferred to another vessel in a new culture medium. This is also known as subculturing. This transfer of cells from one medium to another is known as passage. The cell culture established after first passage is known as secondary culture. The primary and secondary culture contains different types of cells which are derived from the original animal tissue. The cells that adapt to the medium exhibit prolonged growth and are passaged repeatedly. When the cells survive some passages, it becomes a cell line. A cell line develops when the cell grows rapidly for numerous generations. A certain cell type or cell lineage dominates in the culture and exhibits a specific degree of uniformity in the population. Cell lines are usually undifferentiated. They adapt to the culture conditions and lose specific functions and original cell structure. Some cell lines developed from differentiated cells may display characters of differentiated cells. For example, some hepatoma cell lines such as HEPG2 synthesize most of the serum proteins made by normal hepatocytes. Some cell lines survive for limited number of generations and enter into senescence. They are known as finite cell lines. They may survive for 20 to 80 generations. This depends on the cell line type, culture conditions and clonal variation, etc. Cells with some specific properties can be separated from this culture by cloning or physical methods of separation. Such cells growing in a culture are cell strains. The cell line growing in vitro may undergo some changes and multiplies indefinitely. It is known as a continuous cell line. The continuous cell lines grow indefinitely because they have escaped from the senescence control. The cells in the continuous cell line are cloned and characterized for specific applications. Such cell cultures are known as continuous cell strains. The continuously growing cell lines must be examined and characterized thoroughly because many of them get contaminated with fast growing cell lines cultured in the laboratory, such as HeLa cell lines. HeLa cell line is an established cell line developed from cancerous cervical tumor cells of Henrietta Lacks in 1951. They have been successfully subcultured since then and are used in various studies. Some finite cell lines originated from normal tissue are fibroblast cell lines IMR90, MRC5, MRC9, WI38 originated from human embryonic lung tissue. Some examples of continuous cell lines are epithelial cell line 293 originated from human embryonic kidney, fibroblast cell line 3T3A31 originated from mouse embryonic tissue. Some continuous cell lines originated from neoplastic tissue are epithelial cell line A2780 originated from adult human ovary, neuronal cell line C1300 originated from adult rat neuroblastoma, etc. The cells growing in primary culture exhibit properties quite similar to normal cells in the organism. The normal cells do not adapt to the artificial conditions provided in vitro to the mammalian cells. Therefore, these cells stop growing after limited number of divisions. The cells which grow longer in the culture medium exhibit some properties which are different from the characteristics of the normal cells. These different characters are mainly because of a series of changes occurring in a cell culture. These changes eventually lead to the development of established cell lines. They continue to multiply even after a large number of passages and grow indefinitely. Cells in established cell lines are the transformed cells which possess altered characters. Transformation refers to the change in the genetic makeup of the cell which is exhibited phenotypically. The change from normal to transformed cells occurs gradually while growing cells in vitro. Genetic instability in the cells leads to transformation. The cells growing in culture are prone to genetic instability. It is phenotypically expressed in the form of aberrant growth control, immortalization, and malignancy. Normal cell cultures developed from human tissue are generally genetically stable, whereas the mouse cell cultures are genetically unstable. Cell lines developed from tumor cells are genetically unstable. The process of transformation can be spontaneous or induced. It can be induced by chemical carcinogens or oncogenic viruses. A transformed cell in vitro exhibits remarkable difference from the normal cells. Transformation is the process which takes place step by step. There are a variety of characteristics which are exhibited by transformed cells. A transformed cell may exhibit a few or all the characteristics. The features which demarcates normal and transformed cells are contact inhibition, anchorage independence, altered size and shape, aneuploidy, immortality, tumorigenicity, malignancy, and low serum requirement and growth factor independence. Contact inhibition. The cells in primary culture grow in the culture vessel in a pattern similar to their growth in vivo. As the cells are multiplying, they keep on spreading till they come in contact with the vessel wall. As soon as the cells touch the culture vessel wall, they stop growing. This leads to the formation of a single layer of cells on the culture medium, also known as monolayer culture. If the cells get transformed, their growth is not inhibited by reaching in contact with neighboring cells or culture vessel. The transformed cells continue dividing even after attaining confluence and grow in multi-layers instead of monolayer cultures. They have a capacity to grow at higher cell density than normal cells. The cells exhibit increased motility on the loss of contact inhibition, anchorage independence. The normal cells which grow in primary culture require a support for growth. These cells grow in vivo also by adhering to the adjacent cells and tissues through the extracellular cementing matrix. They produce cell adhesion molecules which anchor them to the neighboring cells and substratum in the cell culture. Neoplastic transformation may involve cell surface modification which leads to changes in glycosylation and cell adhesion molecules. They may lose cell adhesion molecules and start growing without the support. These changes may initiate the capacity of cells to grow without the need of substratum, thus increasing motility, which can also be correlated with the development of invasion and metastasis in vivo. These cells can grow in any dimension, and instead of forming layers, they can form aggregates of cells. They can also grow in liquid medium as suspension cultures, altered size and shape. Normal cells have specific size, shape and structure. Cells dispersed from any organism for establishing a primary culture are usually the differentiated cells with definite functions and related morphology. Placing these cells on culture medium diminishes the activity of cells, which may lead to the loss of differentiated features. The cells which undergo transformation lose their original size and shape. The cells lose contact inhibition property and get disoriented. They lose their original shape and exhibit rounded morphology. Aneuploidy. Cell lines do not show genetic stability. When the cells are growing on artificial medium, the chromosome number and content readily changes. Chromosomal aberrations and variations in DNA content are observed because of mutations occurring in the cells. The number of chromosomes is 2n in normal cells. Chromosomal deletion or duplication may lead to heteroploidy of chromosomes. Immortality. The normal cells have finite lifespan, which means they can grow for limited number of generations. The number of generations a normal cell can grow depends on various factors. The cells die due to necrosis or apoptosis after specific number of generations. Mouse cells exhibit changes in the karyotype after 12 generations, which leads to the selection of cells which can grow indefinitely. They easily give rise to continuous cell lines like the tumor cells. The cells which undergo transformation lose the control over multiplication and acquire infinite lifespan. They multiply indefinitely and grow for generations after generations. The lifespan of cells is controlled by a set of genes. These gene products regulate the cell cycle. Immortalization is achieved by inactivation of cell cycle genes, which leads to the loss of control of senescence. Mutation or overexpression of these genes can lead to loss of control of senescence. Changes in oncogenes expression can also lead to the loss of growth control. Immortalization is also related to telomerase activity. Telomerase is an enzyme which synthesizes the lost portion of the telomere. Telomeres are the repetitive sequences at the end of eukaryotic chromosomes, which protects the chromosomes from DNA damage. The cells having finite lifespan do not have telomerase activity, because of which the length of the telomere goes on decreasing. The stem cells and tumor cells possess high telomerase activity, whereas normal and differentiated cells possess very less or no activity. Immortalized cells may lose the ability to differentiate. Cells can also gain immortalization due to the interference of viral genes. Immortalization is also induced in the cells by transfecting telomerase gene. It plays essential role in chromosome stability and cellular lifespan. The cell lines which enter into immortalization may retain contact inhibition, anchorage dependence and tumorigenicity. Tumorigenicity. Tumorigenicity is the ability of the cells to form tumors in vivo or in vitro. Cell lines derived from tumor cells attain tumorigenicity faster. They exhibit infinite lifespan and are further transformed to increased growth rate, reduced anchorage dependence, more pronounced aneuploidy and immortalization. All the cells in a given tumor may not have the same transformed characteristics. The transformed cells, when injected into an experimental animal, develops a tumor, whereas a normal cell does not. Malignancy. Malignancy is the property of cells to invade other tissues of the body and start proliferation elsewhere to establish tumor. Malignancy is usually associated with uncontrolled growth and spreading of multiplying cells in other tissues in vivo. It normally leads to the loss of density limitation for cell proliferation and motility control. Loss in motility control means loss of contact inhibition, where the cells keep on growing even when the space is not available. Low serum requirement and growth factor independent. The cells in culture require serum for vital metabolic activities of growth because it contains lots of growth factors, whereas tumor cells can produce the required growth factors on their own, hence do not require the external supply. Transformed cells exhibit lower serum dependence because they synthesize adequate amount of growth factors required for the growth. Tumor cells are known to produce many hematopoietic growth factors such as interleukins 1, 2, 3 and colony stimulating factor. Growth curve. Cells in the culture medium exhibit a specific pattern of growth. The growth curve of normal cells in culture medium can be depicted in the form of a sigmoid graph. Cells display following phases of growth in the culture medium: lag phase, log or exponential phase, stationary or plateau phase, senescence or death phase. Lag phase. The cells after inoculation in the fresh medium do not start multiplying immediately because they adapt to the new environment. The cells transferred into the new medium are detached from the previous culture vessel with the help of enzymes. This treatment leads to the loss of extracellular matrix of the cells, which helps them to attach to the substratum. Cells synthesize glycocalyx and spreads on the substratum with the help of cytoskeleton. There is nearly no increase in the cell number in lag phase. The DNA polymerase activity increases and the new proteins are synthesized. The time taken by the cells to complete lag phase depends on the phase of growth from which cells were picked up, the concentration of cells in the inoculum. The cells picked up from the actively dividing phase exhibit shorter lag phase, log phase. The cells divide exponentially in the log phase. Logarithmic or exponential phase is the best stage for studying cell activities, as most of them, that is around 90 to 100%, are in active physiological and dividing phase. Cells in the log phase are more consistent and uniform. Growth of cells in log phase is influenced by inoculum concentration, nutrient availability and accumulation of toxic metabolites. The response of cells in culture can be studied well in this phase, such as effect of nutrients, hormones, toxic drugs, etc. This is the most reproducible stage and appropriate for sampling. The cell population is uniform and the viability is high here. The cells may be at different stages of cell cycle. The cell doubling time, also known as the population doubling time, can be determined in this phase. The population doubling time is different from cell cycle or generation time because it is the average of the time taken to double the population of cells in the given culture. The cells in the culture may take varied time interval to divide. The cell cycle time or generation time is measured from one point in the cell cycle until the same point is reached again. Population doubling time is influenced by non-growing and dying cells also. It may range from 12 to 15 hours in rapidly growing mouse leukemias to 60 to 72 hours in slow growing finite cell lines. The log phase comes to an end when the cell concentration exceeds the capacity of the culture medium. Log phase may end for the adherent cells when they attain confluence due to contact inhibition. At this stage, the medium must be changed or culture must be divided into separate vessels. The adherent cells are subcultured by dissociating the cells from the culture vessel. The culture medium is removed and enzymes like trypsin, pronase, dispase and collagenase may be added for dissociation of cells. Stationary phase. The culture attains confluence by the end of the log phase. The cells cover the entire surface available in the culture vessel and all of them are in contact with each other. Growth rate reduces after achieving confluence and eventually ceases. The cessation of growth due to confluence is termed as contact inhibition, which leads to the formation of monolayer. Epithelial and endothelial cells form a monolayer in the culture medium. The growth fraction falls between 0 to 10. Cells in the plateau phase are more differentiated and have reduced growth fraction. They secrete more intercellular matrix and are therefore difficult to disaggregate. The protein synthesis shifts from structural to specialized. The cells become less motile. Growth of cells also slows down due to the accumulation of toxic inhibitory metabolites. Depletion of nutrients and growth factors also leads to reduced cell spreading. Cell division and cell death becomes equal, which leads to the stationary phase. After certain time period, the cell division completely ceases. The stationary phase can be prolonged by replenishing fresh culture medium. The cells which get transformed continue dividing even after attaining confluence and reach higher cell densities than their normal counterparts. The cells dividing in this case compensates for the loss of cells dying in the plateau phase. Such cells are anchorage independent. Death phase. At the end of stationary phase, death phase starts. Here, the dying cells are not replaced by the dividing cells and the number of viable cells decreases. The cell death may occur due to necrosis or apoptosis. If the culture constitutes of normal cells, most of them die, but if the culture is of transformed immortalized cells, they soon start growing actively on subculturing. Cells follow the standard growth pattern when subcultured in a fresh medium. The animal cells grow in the culture initially in a pattern similar to their growth in vivo, but as they adapt to the artificial cultivation atmosphere, they may undergo some changes and lose the normal characteristics to become established cell lines with specific growth patterns.